Imagine a massive earthquake hits a sprawling, highly developed city. Buildings collapse, bridges fracture, and the infrastructure is, well, it's completely
Yeah,
compromised.
yeah, total chaos.
Right. And the city needs to rebuild, and it needs to happen fast. City planners, they don't sit around waiting for new steel to be forged in some distant factory or for fresh timber to be cut and shipped from across the country.
No, that takes way too long.
Exactly. The city is vulnerable to aftershocks right now. So instead, they send crews directly into the rubble. They salvage the intact bricks, pull out the steel beams that are still structurally sound, and immediately use those exact same materials to patch the critical infrastructure.
You bypass the supply chain delays, basically, because the emergency is already happening.
Mm.
Every second you wait for new materials is, a second, and the entire system is at risk of total collapse.
And that exact same emergency recycling is happening inside your cells right now. When your DNA snaps, your cells don't wait for new materials, they scavenge their own wreckage. So, welcome to today's deep dive. We are looking at how the human body runs this ultimate microscopic recycling program.
Yeah, the enzymatic salvage pathway.
Right, the enzymatic salvage pathway. It's the way your cells recycle degraded genetic material into free nucleotides for rapid, life-saving DNA repair. And we've got a great stack of sources for you today, including biochemistry textbooks, oncology journals from PLOS1 and AACR, and, well, one really unexpected source.
Oh, yeah, the catalog.
Yeah, I found myself reading through a commercial shopping catalog for scientists from a company called MyBiosource. Okay, let's unpack this because it blew my mind. It's basically a hardware store catalog for molecular biologists.
It really is. It lists the exact recombinant proteins and antibodies, like the actual molecular tools that scientists are actively buying to track this recycling process in the lab.
Which made me realize that cellular recycling isn't just some minor metabolic efficiency upgrade. It is a massive, heavily researched emergency supply chain for our genome.
Absolutely. And to really understand the stakes of that supply chain, we have to look at the cellular economy. There are fundamentally two ways a cell can acquire the nucleotides it needs to build or repair DNA and RNA. The first is called de novo synthesis, meaning from scratch.
Which is the equivalent of forging new steel and cutting fresh timber in our city analogy.
Exactly. And industrial manufacturing is incredibly energy intensive. To build a purine ring, those are your adenine and guanine bases, the actual letters of your genetic code. The cell has to assemble it step by step.
It's not a quick process.
Not at all.
Yeah.
It has to scavenge amino acids, pull in carbon dioxide, and just burn through massive amounts of ATP, which is the cell's main energy currency. The entire structure is slowly built directly onto a sugar phosphate scaffold.
And it's similar for the other bases too, right?
Yeah, pyrimidines, urethymine and cytosine, are also built from scratch, though the ring is constructed first as a free base before being attached to the sugar. But the sheer energetic cost of this process is staggering. I mean, the cell is pouring its energy budget into creating just a single building block.
Okay, wait, I have to push back here. If de novo synthesis is so incredibly expensive and energy draining, why does the cell even bother doing it? Why not always just salvage everything and save the energy?
Well, the answer comes down to volume versus efficiency. Think about a cell that is rapidly dividing.
Like an embryonic cell.
Right, or the cells lining your gut. When a cell divides, it has to duplicate its entire genome. That's 3 billion base pairs.
Oh.
The sheer volume of nucleotides required for that complete replication far exceeds what a cell can scavenge from its own internal recycling bin. There just isn't enough rubble lying around to build an entire second city.
Okay, that makes sense.
So for that massive demand, you need the heavy industry of de novo synthesis. But for daily maintenance, routine wear and tear, and localized damage, the salvage pathway is highly efficient and absolutely essential.
So let's look at how this renovation actually works on the ground, molecularly speaking. How does the cell actually harvest the rubble?
It starts with the breakdown of existing, maybe damaged, nucleic acids. Polynucleotides, which are long chains of DNA or RNA, are degraded into smaller pieces. But immediately we hit a logistical hurdle.
The phosphates.
Yep, nucleotides have a phosphate group attached to them. That phosphate is bulky and it carries a strong negative charge.
And cellular membranes, the lipid bilayers that separate different compartments in the cell, are highly hydrophobic. They strongly repel charged molecules.
Right.
So trying to push a highly charged phosphate group through a lipid bilayer is like trying to push a giant waterlogged sponge through a thick wall of oil. It bounces off.
That's a great way to picture it. So to transport these vital components across cellular compartments, the cell has to modify them. It uses specific enzymes called nucleotidases to surgically strip away that bulky, charged phosphate group.
And that changes the charge.
Exactly. Stripping the phosphate converts the nucleotide into a neutral molecule called a nucleoside. Once it is neutral, it can easily slip right across the lipid bilayer to wherever it's needed.
Which is where that My Biosource catalog becomes so relevant. Reading through the listings, you see researchers constantly ordering things like cytosolic 5' nucleotidase 1A.
Or NT5C1A for short.
Right. They're buying in antibodies to track this exact stripping process in real time. They're also buying purine nucleoside phosphorylase, or PNP, and guanine diminase. These are the specific molecular demolition crews breaking down the recycled goods so they can be transported.
And once those free bases are safely transported and ready for reuse, they need to be rearmed. A neutral nucleoside can't be stitched into DNA. It needs its phosphate back.
So how does it get it?
They meet up with a prefabricated sugar phosphate scaffold called PRPP. Then, specialized salvage enzymes step in to finish the job. An enzyme called APRT handles adenine, and one called HGPRT handles hypoxanthine and guanine.
And they just snap them together.
Yep, in a single, highly efficient step, they snap the recycled base directly onto the PRPP scaffold. You have a fully functional, ready-to-use nucleotide, entirely bypassing all those costly, slow de novo assembly steps.
Okay, so we've established that the salvage pathway is this brilliant economic lifesaver for the daily running of the cell. But, and here's where it gets really interesting.
And things go wrong.
Yes. Diving into the PLFS1 study, it becomes clear that this exact speed and efficiency become a literal life-saving tool during a genetic crisis. The study focuses on double-strand breaks, or DSBs. That's when both strands of the DNA double helix, both rails of the ladder, are physically snapped.
And a double-strand break is the cellular equivalent of a 5-alarm fire. When only one strand breaks, the cell can use the intact opposite strand as a template for a quick fix. But when both strands snap, the structural integrity of the chromosome is just gone.
That sounds bad.
It is. If it isn't repaired quickly and accurately, the cell will undergo apoptosis that's programmed cell death, or even worse, the broken ends will fuse improperly, which can potentially lead to cancer.
Right. And the most accurate way the cell fixes this is through a process called homologous recombination, or HR. The PLOS1 paper points out that HR is highly effective, but it requires extensive new DNA synthesis to fill in the missing gaps at the break site.
It demands an immediate massive flood of nucleotides, specifically the building blocks known as DNTPs.
So if the DNA is physically snapped in half, how long does the cell actually have to flood the site and fix this before it dies?
It's a terrifyingly short window. The PLOS1 research shows that the vast majority of this critical DNA repair must happen within the first 8 hours after the genotoxic stress.
Wow, 8 hours. And that stress could be anything, right?
Yeah, that genotoxic stress could be a dose of radiation, exposure to a chemical toxin, or even just a severe metabolic error.
And the de novo synthesis assembly line is way too slow to power up and deliver in that 8-hour window. The cell absolutely has to rely on the salvage pathway. But how does the recycling center know that a catastrophic earthquake just happened? How does it know to switch into absolute overdrive?
It relies on a brilliant biochemical signaling mechanism. When a double-strand break occurs, a sentinel protein called ATM detects the severed DNA ends immediately. ATM is a kinase.
Like an emergency dispatcher.
Exactly. ATM sounds the alarm. One of its direct molecular targets is a salvage enzyme called DCK, or deoxycytidine kinase. When ATM detects the break, it rushes over and phosphorylates DCK.
Meaning it adds a phosphate group?
Right. It attaches a phosphate group to a very specific spot on the DCK protein serine 74. And this isn't just a chemical tag. It physically forces the DCK enzyme to change its three-dimensional shape.
Oh, that's cool.
Yeah. And this shape change acts kind of like a molecular turbocharger. It drastically increases DCK's activity, causing it to just churn out massive quantities of the nucleotide DCTP, which is desperately needed at the site of the break for homologous recombination.
And the researchers actually proved this turbocharger effect was the key to the 8-hour window, right? They went in and mutated that specific spot on the DCK enzyme that serine 74, so ATM couldn't attach the phosphate.
Yep.
And when they blocked that single communication line, the repair rate just plummeted and the cells couldn't survive the damage.
It beautifully illustrates how the salvage pathway isn't just a passive system running in the background. It is dynamically regulated by the cell's damage response network.
Which is amazing.
And the sources also highlight another crucial salvage enzyme involved in this targeted response, which is thymidine kinase 1, or TK1.
Oh, right, the Comet FIsh assay stuff.
Yeah, using a technique called a Comet FISH assay. which let scientists visually track DNA repair in specific individual genes under a microscope, the research showed something remarkable. TK1 is strictly required for the preferential repair of actively transcribed genes.
So genes that are actively being used, the ones most critical for whatever that specific cell's current job is, get VIP treatment from the recycling crew.
Exactly.
The cell prioritizes its repair efforts, sending the TK1 salvage enzyme to deliver materials directly to the most vital active areas first.
It's triaged on a molecular level.
But wait, if you are flooding the disaster site with millions of recycled bricks in a complete panic to meet this 8-hour window, aren't the builders going to start making sloppy mistakes? I mean, if there is just an artificial overabundance of building materials floating around the break site, doesn't that increase the chance of genetic errors?
This raises an important question, and it's a fundamental paradox of DNA repair. The source material, particularly studies looking at yeast models during oxidative stress, confirms exactly what you were asking.
So it does cause errors.
Yes. Enlarging the DNTP nucleotide pools is absolutely essential for effective DNA repair and cell survival. If a cell cannot rapidly expand its nucleotide pool through salvage, it just dies from the unresolved DNA breaks. However, that exact same enlarged pool directly results in higher mutation rates.
Because the machinery gets overwhelmed.
Pretty much. The repair polymerase, the molecular machines that actually stitch the new nucleotides into the broken DNA string, they operate based on binding affinity and kinetics.
Meaning they rely on the right pieces floating by at the right speed.
Right. When nucleotides are overly abundant, the polymerases start working too fast. They become less selective. The local concentration of nucleotides is so high that the wrong base can easily slip into the active site of the polymerase and get permanently incorporated into the DNA sequence.
It's a classic biological trade-off. You ensure survival today by patching the hole, but you potentially introduce a mutation that causes problems tomorrow.
And the cell is exquisitely aware of this danger. It constantly monitors the size of its nucleotide pool to manage this risk. It uses an enzyme complex called ribonucleotide reductase, which has a specific allosteric site, like a sensory switch.
What does the switch do?
This switch continuously monitors the ratio of ATP to DATP in the cell. By constantly weighing the concentration of cellular energy against the concentration of raw DNA building blocks, the cell tries to walk a tightrope. It attempts to provide just enough salvage materials to survive the immediate, lethal threat of a double-strand break without saturating the site so heavily that the polymerases cause massive cancer-causing mutations.
Knowing that this balance between immediate survival and long-term mutation is so incredibly delicate, it makes you wonder what happens when this cellular recycling system breaks down completely.
It's not good.
Or what happens when a specific tissue in the human body lacks the heavy de novo machinery and is completely reliant on salvage.
The consequences are severe. While the liver operates as a massive de novo synthesis factory, constantly churning out new materials, certain tissues lack that robust machinery entirely. The brain, the bone marrow, and mature red blood cells, erythrocytes, are fundamentally dependent on the salvage pathway.
I was reading the section on erythrocytes, and it is fascinating. Mature red blood cells don't even have a nucleus. They expel it during development to make more room for oxygen-carrying hemoglobin.
Yeah, they're basically just bags of hemoglobin.
Exactly. They do not synthesize new DNA or RNA at all. So why do they need nucleotides?
They require a constant supply of nucleotides for their basic energy metabolism. ATP, the energy currency we've been discussing, is a nucleotide. Red blood cells need it to maintain their shape, manage their ion channels, and survive their journey through the circulatory system.
And they can't build it themselves.
Right. Because they cannot build anything from scratch, they get their building blocks exclusively by salvaging preformed purines that are synthesized and shipped out into the bloodstream by the liver.
They are entirely dependent on that metabolic supply chain. But the brain is perhaps the most striking example of what happens when salvage fails. The biochemical texts discuss Lesch-Nyhan syndrome.
Yes.
It's a devastating real-world pathology caused by a genetic defect in just one single recycling enzyme, HGPRT.
The brain is heavily protected by the blood-brain barrier, and neurons are massive energy consumers. The energetic cost of running de novo synthesis is simply too high for neurons to sustain while also managing their electrical firing. They rely heavily on salvaging purines to meet their metabolic needs.
And because they rely so heavily on that salvage economy, losing that one HGPRT recycling enzyme doesn't just cause a minor metabolic hiccup. It leads to severe, debilitating neurological symptoms. Patients with Leschnahan experience severe gout, poor muscle control, cognitive impairment, and a tragic, compulsive, self-injurious behavior.
Heartbreaking.
It really proves that this enzymatic salvage pathway isn't just a biochemical curiosity. It is a foundational pillar of human neurobiology and health.
It underscores the absolute necessity of the pathway. But if we connect this to the bigger picture, as we discussed with the mutation risk at the disaster site, having the pathway constantly turned on, constantly flooding the cell with materials, is also highly dangerous.
Right. You need a way to stop it.
Exactly. The cell needs a way to stop the salvage pathway once the emergency is handled. It needs a brake pedal. The sources point to a fascinating protein called SAMHD1 that acts as a master regulator.
The text describes SAMHD1 as having DNTP triphosphohydrolase activity.
Which is a complex way of saying it is an enzyme that actively seeks out free, ready-to-use nucleotides and destroys them.
Oh, wow.
Yeah, it strips the phosphates off, degrading the triphosphates back into simple, unusable nucleosides and inorganic phosphate. SAMHD1 is the brake pedal. By steadily degrading the intracellular nucleotide pool, it actively lowers the global concentration of building blocks in the cell.
And that prevents the overaccumulation that causes the repair polymerases to become sloppy and introduce errors. But wait, if the cell just survived a massive double-strand break by utilizing the salvage pathway to get materials, why would it deploy a protein that destroys those exact same building blocks?
What is truly remarkable about SAMHD1, and this is a relatively recent discovery highlighted in the literature, is that it is not merely a passive regulator lowering the overall pool size. It actively participates in the repair of the double-strand breaks.
Wait, the protein that destroys the bricks is also helping fix the collapsed building?
It seems completely counterintuitive, but SAMHD1 actually localizes directly to the site of the DNA damage. Once it arrives at the break, it facilitates homologous recombination by physically recruiting other essential repair proteins.
Like a site manager.
Yes, it brings in factors called CTP and MRE11. You can think of these factors as the specialized demolition and prep crew. They are required to trim back the jagged, broken ends of the DNA so the repair polymerases can actually start their work.
Okay, so SAMHD1 is lowering the global amount of bricks in the city so people don't trip over them and make mistakes. But at the exact site of the collapsed building, it is acting as the site manager. It's coordinating the specialized prep crews to clear the jagged rubble and get the site ready for the salvage materials.
That's a perfect analogy.
That is an incredibly sophisticated level of multitasking for a single protein.
The pathways of synthesis, degradation, and repair are deeply intertwined. The cell doesn't view them as separate departments. They're all part of a single, fluid response to genotoxic stress.
So what does this all mean? When you put all these pieces together from the textbooks, the oncology journals, and even the MyBiosource catalog, the picture totally changes. The cellular salvage pathway isn't just an effort to save a few molecules of ATP.
Not at all.
It is a highly regulated, rapid response emergency protocol. Utilizing specialized enzymes like HGPRT, DCK, and TK1, your cells can instantly harvest their own molecular trash, strip it down, rearm it, and stitch your genome back together in a matter of hours.
All while being managed by regulatory proteins like SAMHD1 to ensure that this frantic repair doesn't accidentally cause cancer-causing mutations.
It is a continuous, dynamic negotiation between energy conservation, genomic stability, and immediate survival. It really puts things into perspective. Next time you step out into the sun and get a little UV exposure, or honestly just go about your normal day breathing oxygen and metabolizing food, remember that your DNA is constantly taking hits.
It's constantly fracturing.
But your microscopic recycling plant is working overtime. It is actively scavenging the wreckage to patch up your genome and keep you functioning.
The sheer scale and speed of the invisible work happening inside our cells to keep us intact is staggering.
It really is. But I want to leave you with a final thought based on everything we've unpacked today. Something for you to mull over. We know that cancer cells divide incredibly rapidly. They have an insatiable hunger for raw materials, especially nucleotides, to copy their mutated genomes.
Right.
And we know from the AACR Oncology Journal that cancer cells rely heavily on these exact salvage pathways to survive the intentional, massive DNA damage caused by chemotherapy drugs like osimertinib.
They essentially hijack our body's natural emergency response system to save themselves from the medical treatment.
So what if we use that against them? What would happen if we designed drugs that completely turned off their recycling plants? If we could temporarily disable salvage enzymes like DCK or PNP in a tumor while simultaneously hitting it with a chemotherapy drug that causes massive double-strand breaks, Could we trap the cancer cells in their own rubble? Could we create a scenario where their DNA is shattered? They desperately need to rebuild within that 8-hour window, but we've completely cut off their access to the salvaged materials they need to survive.